Hi everyone. Today in this lecture I am going to make you understand about the enzyme substrate interaction and what are the models that were used to explain the enzyme substrate interaction, and along with that we will also look into the factors affecting enzymes. Right, so we start. So we know that every enzyme is actually a protein, right? And enzymes are those proteins, these are actually globular proteins, right? And they in their tertiary structure um, have some adjustments. Proteins in their tertiary structure, when they have certain adjustments, right, these lead to formation of enzymes, right? So every enzyme contains within its tertiary structure cluster of amino acids that forms the active site, right? So contains within its tertiary configuration, which means that enzyme is a protein, and if you know, protein has three main structures. These are primary, secondary and tertiary. So tertiary structure of the protein is a more complex arrangement where all the amino acids, they come closer to each other for better arrangement, right? And since enzymes are proteins, so we are talking about its tertiary configuration, right? So every enzyme in its tertiary configuration has cluster of amino acids. Since enzymes are proteins, these will be made up of amino acids, and therefore at the active site of the enzymes, cluster of amino acids are present, and these amino acids makes the active site of the enzyme, and this is the site where the catalytic event takes place. That is, this is the site where the reaction process takes place, where the substrate binds to the enzyme, and this leads to formation of the product, right? So the active site is the one where the catalytic event takes place. So and the catalytic event that we are talking about is a reaction for which the enzyme is responsible. So every enzyme is responsible for different reactions. Every enzyme speeds up different reactions. So the enzyme, whatever may be the enzyme, is responsible for binding of substrate, and this leads to catalytic event, which is the reaction for which the enzyme is responsible, right? So the active site that we are talking about is an actual groove or pocket. So active site is a pocket like structure, right? And where it has its own chemical structural properties, right? So this pocket this pocket, which is the active site, accommodates the substrates, right? And the enzyme is of high specificity, right? So not all substrate can bind to the active site of the enzyme, because enzymes are substrate specific, right? So active site is a very important part in the structure of enzymes when we come to enzyme substrate interaction. So one more thing that you might be thinking, that what is catalytic event? So it is basically the process of catalysis. This is the process of increasing the rate of a chemical reaction, right, by adding something, right? So catalysis can happen by adding a substance like enzyme, a substance like inorganic catalysts, right? Or there are times when reaction do not have catalysis also, right? So the work of enzyme is mainly to increase the rate of that reaction, and that is why it is mentioned here that the active site is the place where the catalytic event takes place, right? So now I hope you all know that there are 20 different kinds of amino acids, and all these amino acids helps in making the proteins, right? So though there are 20 different types of amino acids, not all of these are involved in the formation of active sites. We already discussed that active sites are made up of certain amino acids, but not all of these 20 amino acids that we know gets involved in the formation of active site. So the main amino acids, then we need to know that what are the main amino acids that are actually involved in the active site formation. These are cysteine, serine, histidine aspartate, glutamate and lysine. So mainly these 6 amino acids are the amino acids that are associated with the active sites. These participate in binding the substrate to the active site. So when the substrate has to bind to the active site, these are the amino acids that are present for the substrate binding at the active site, right? So and also of these 6 amino acids, histidine, aspartate and glutamate, these 3 amino acids act as donors or acceptors of proton, right? Sometimes these also act as donor or acceptors of proton. We need to know that to carry out reactions, that is oxidation takes place, and for that we need to there has to be donation or acceptance of protons, and this is these 3 enzymes work in that direction. Now we saw that at the active site amino acids are present. These 6 amino acids are present. Not all these 6, but depending upon the enzyme, the amino acid present out of these 6 may vary. But in addition to these amino acids, enzyme also have non-protein components. So non-protein components means that these are not enzymes. So apart from enzymes, there are also certain non-protein components present which are not enzymes, and such groups are called prosthetic groups. Prosthetic groups are those that help enzyme in carrying out the reaction, but these are non-protein, these are not enzymes, right? And these prosthetic groups are actually small organic molecules, say for example iron or zinc. So usually these prosthetic groups are present which act as acceptors of protons, right? So what actually happens is that amino acids, the side chain amino acids are not good acceptors of donors, acceptors of proton. So prosthetic groups act as acceptors, and in this way they help the reaction to proceed, right? So at the active site, remember that there are certain amino acids present, and along with the amino acid also a prosthetic group is present which is not a protein, right? Now because of the chemical fit between active site of an enzyme and its substrate, enzymes are effective as catalysts, right? So as we know that enzyme and substrate, they have a proper chemical fit, they fit with each other, and therefore enzymes are effective as catalyst as enzyme and substrate combines with each other. This forms enzyme substrate complex, and therefore enzymes are effective as catalysts. Catalysts means those substances that can carry out the process of catalysis. Now the enzyme catalysts can actually proceed 10 raised to 7 to 10 raised to 14 times faster than uncatalyzed reactions. So as I told, catalysts are something that speed up the rate of reaction. So in comparison to an uncatalyzed reaction, that is a reaction that in which catalysis is not taking place, right? In comparison to such reactions, reactions that are catalyzed by enzymes proceeds much faster. The rate of reaction is much faster, that is 10 raised to 7 to 10 raised to 14 times faster than the uncatalyzed reactions. Whereas enzyme catalyst, the reaction that is catalyzed by enzymes proceed 10 raised to 3 to 10 raised to 4 times faster than inorganic catalysts, right? So enzyme is not an inorganic catalyst, but in comparison to inorganic catalyst, enzyme catalyst can proceed faster, right? And therefore enzymes are always known as biocatalyst, because they carry out most of the biological reaction at a very great speed, right? Now let us have a look into the process of substrate binding. So what happens initially? What is the initial way by which the substrate comes into contact of the enzyme? So substrate, for those who do not know, substrate is the substance that will combine with enzyme to give the product, right? So the initial contact takes place between the enzyme and the substrate because of random collisions. So randomly there are collisions in the medium, and these collisions lead to the interaction of enzyme and substrate, right? After this random collision, once the substrate is in the groove of the active site, right, so there the substrate will bind enzyme surface temporarily. So after the collision, due to those random collisions, the substrate will come in contact with enzyme, and once it is in the contact of enzyme, once it is in the groove, the pocket of the active site of the enzyme, this substrate will bind enzyme surface temporarily. By temporarily, I mean that weak bonds will be formed. These weak bonds are hydrogen bond and ionic bond. These are weak bonds. These can be broken down easily. So that is why it is mentioned that substrates bind enzyme surface temporarily. As these are weak bonds, they can be broken down easily, but these bonds are present many in number, and therefore this holds the enzyme substrate complex in a place. So this is the initial part when the substrate binds, right? Then okay, so now let us take a look into the way in which enzyme and substrates bind. So there are two models that explain the enzyme substrate interaction, that is the way in which the enzymes interact with the substrate. These are Lockin Key Model and Induced Fit Model, right? So let us first take a look into Lockin Key Model. Now this model was proposed by Emil Fischer in 1894. So Emil Fischer is the scientist who proposed this Lockin Key Model, and this model explains that active site complements the substrate precisely, which means that active site shape is exactly in such a way that substrate can exactly fit into it. That is, the shape is completely complementary, so that both of these can fit into each other, right? And Therefore this model is called Lockin Key Model, because it's like lock fits into key. In that way, this is called Lockin Key Model. This model explains the enzyme, that the enzyme exhibits specificity. We will look into it in the next slide, but this model gives you an idea that enzyme are substrate specific, and there takes place no conformational changes in the shape of the enzyme. So this model says that there is no conformational change in the shape of the enzyme, right? Now let us come into the induced fit model. Now this model is proposed by Daniel Koshland in 1958. So Daniel Koshland is the scientist who proposed induced fit model, and he proposed this in 1958. Now this model is on contrary to Lockin Key model. This explains that active site is not completely a rigid fit to the substrate. So this model tells that active site is not exactly complementary to the substrate. The shape is not exactly complementary to the substrate. So this model explains that enzyme exhibit broad specificity. This model gives us an idea that the enzymes are broadly specific. So they can bind to some certain substrates. One enzyme can bind to, like, say 8 or 9 substrates, right? So it says that enzymes are broadly specific, and this model believes that conformational changes take place when exposed to substrate. So this model says that in the structure of the enzyme in the active site, the conformational changes take place. Conformational changes means changes in the tertiary structure of the enzyme. So as I told that enzymes are proteins, they have tertiary structure. So adjustments take place in the tertiary structure of the enzymes, and this is what is supported by induced fit model. But the Lockin Key model says that there is no conformational change that is taking place. So let us understand each of these models in a bit more detail. So let us take a look into Lockin Key model. So in this model, as I told, active site was thought as a rigid structure. So by rigid structure we mean that the site is not going to change its shape. There would be no change in the shape of the active site. So it is a rigid structure, and the substrate will fit exactly into the active site. Since there is no change in the structure of the active site, the substrate would be specific, and only those substrate that can fit into the active site, into the structure, will fit in the active site. So this model tells that substrate is like a key and enzyme is like a lock, right? So in the lock, which is the enzyme, the right key, the right substrate which can fit into the lock, right, will only bind to that enzyme. And we know what happens in lock and key, that only that key will open the lock which is right for that lock, right? And therefore this is called the Lockin Key model. So which means that both of these, the enzyme and the substrate, they have the complementary shape. Now as I told, this model explains the enzyme specificity. So this as I told that since substrate are like key, right, so all keys are not going to open the lock, all keys are not going to fit into the lock which is enzyme. So only that substrate which is having key structure, like a key structure that could fit into the so-called lock which is enzyme, only that substrate will be able to bind to the enzyme, and therefore this explains the enzyme specificity, right? But this model fails to explain catalytic event. This model does not give any idea of how the event of catalysis is taking place. So always remember how a Lockin key works, and that is how the Lockin key model is supposed to be, right? So we know that locks have a keyhole, right? And this keyhole, imagine that this keyhole is the active site of the enzyme. So lock is the enzyme, the keyhole that is present is the active site, and the key that we use is the substrate. So only that key which can match the keyhole of the active site will be able to bind to the enzyme. So this is like the correct key that can open the lock. So only a correct substrate will be able to bind to the enzyme because of the correct shape. Now this Lockin key model in this Lockin key model, one problem is there that according to since as this model is the Lockin key model, so inhibitors can also act as a key. So there are certain inhibitors which can have the same shape similarity like the substrate, and these inhibitors can act as a key and hence jamming the active site, right? So in this way there are some flaws in Lockin key model, and actually later on this model was rejected and the model that was accepted was the next model which is induced fit. So what happens in Lockin key model is that suppose this is an enzyme and this is your substrate, right? So both of these have complementary shape. So you can see the shape of the enzyme active site, it complements the shape of the substrate. This leads to formation of enzyme substrate complex. So this is the substrate which gets fits into the enzyme, and this leads to the formation of enzyme substrate complex, and the process of catalysis take place at the active site, and this leads to formation of enzyme product complex. So the enzyme speeds up the rate of reaction, and this leads to formation of enzyme product complex, right? And this leads to then the formation of enzyme giving rise to two products P1 and P2, right? So what is happening that the substrate is going through a reaction where the substrate is getting converted to products, and this reaction is catalyzed by enzymes, where enzymes bind to the substrate, increasing the rate of reaction, leading to formation of enzyme product complex, and then later on releasing the products of interest, right? So this is Lockin key model. The substrate exactly fits into the enzyme as both of these are complementary shape. This was one of the model. Now let us move to understand that what is the induced fit model. So in induced fit model, the enzymes changes its structure. So induced fit model very clearly says that enzyme is not a rigid structure and it changes its structure when the substrate binds. So the initial binding distorts both the enzyme and the substrate. So this model explains that the initial binding can distort. By distort I mean can bring changes in both enzyme and the substrate. So what happens when there is distortion of enzyme? So distortion of enzyme will bring conformational changes of enzyme, right? So by distortion, that is change in the shape of enzyme, we mean that the conformational configuration, the tertiary configuration of the enzyme will be changed. So there would be adjustments, and hence there would be change in active site. Since active site is a part of the enzyme itself, so any change in the conformational change of enzyme means that there would be change in the active site, and this positions the reactive groups in a way that can enhance the reaction. Now this adjustment is mainly for enhancing the reaction, so that the reactive groups that are present can be positioned properly, so that the reaction proceeds properly, right? So that's why this torsion of enzyme is important. Similarly, distortion of substrate puts the covalent bonds under stress. Now covalent bonds are a bit strong bonds, so these bonds has to be broken so that the substrate can be split to give product, right? So the distortion of substrate puts the covalent bonds under stress, and due to this stress the bonds become more favorable to catalytic attack. So when this stress happens, so these covalent bonds can be broken down by catalytic attack, and this will also favor the reaction, right? Now therefore this model says that enzyme is a flexible structure, right? So the active site of the enzyme is a flexible structure where conformational changes take place in the arrangement of the amino acids in the active site. So let us look into this model. So this is enzyme. Now see the shape of this, the active site of the enzyme. It is different than the substrate, right? So as the substrate comes near the enzyme, this will enter into the active site of the enzyme. Still the shape is different, right, of the active site. It is not complementary. But as the catalysis event will take place, you will see that there is change in the structure of the enzyme, and this change makes the substrate to fit properly in the enzyme. So this change is mainly for what? This change is to properly bind the substrate with the enzyme. So first of all, there was the structure was not complementary, but later on there comes changes in the active site due to adjustments of amino acid sequence, and this leads to formation of enzyme product complex where the substrate binds properly with the enzyme and leading to the formation of product. Now then afterwards the enzyme will release the products. So these are the products P1 and P2, and this active site will come back to its usual adjustment, and it will come back to its normal shape, and therefore then it can proceed to another reaction. Now see, this enzyme explains broad specificity. See the active site. It is different than the substrate, right? So any substrate can actually come into this enzyme, right? And later on the enzyme can change its shape according to the shape of the substrate, and this actually explains the broad specificity. That means many types of substrate can bind to the enzyme, which was not possible in the Lockin Key model, right? So this is the induced fit model. Now what happens actually, that as the substrate binds, the conformational changes in the enzyme active site take place. So the change that we are talking about in the active site of the enzyme takes place as the substrate binds. So the substrate has to bind to the enzyme, and then that change in the active site will take place, and this change is for the better interaction of enzyme with the substrate. These changes take place so that the enzyme can better interact with the substrate, leading to the formation of better reaction, right? So what is this conformational change that we are talking about? Conformational change, by that we mean bringing into the active site certain amino acids chain. So certain amino acids chain are brought into the active site, right? And this is critical for the catalytic process. So for the process of catalysis to proceed, this bringing of amino acids into the active site is very important, because these amino acids help in the catalysis process. But to be noted that this amino acid chain that is brought in an induced enzyme is not present in the active site of uninduced conformation. So to be understood that before the substrate binds, the enzyme is an uninduced conformation. The enzyme is not induced. By induced we mean that it is not triggered to carry out the process, right? So it will be induced to bring conformational change only when the substrate binds. So by uninduced conformation I mean that those conformation in active site which has not changed, which has not induced, because substrate has not bind to it, right? So in uninduced conformations active site, that is in uninduced enzymes active site, these amino acid chains are not present in near the active site. So it actually has to be brought to the active site so that the catalytic process can take place. So now let us understand that why it is called induced fit model, because the substrate binding induces conformational change in enzyme. So as the substrate binds, the enzyme knows that conformational change has to be brought, and this triggers to bring the conformational change, and this is important for better fit of the substrate with the enzyme. So this causes better fit, and this is important for the reaction to proceed. So let us understand this with an example of enzyme, which is carboxypeptidase A, right? So carboxypeptidase A, first of all we should know that what is this enzyme. This is an enzyme that breaks down polypeptide by removing one amino acid at a time. So what is the work of this enzyme? It breaks down polypeptide chain. What is polypeptide chain? When many peptides are joined together, it is called polypeptide. That is, it is a sequence of amino acid, but this is a polypeptide since many peptide chains are joined together. So the work of this enzyme is to break down that polypeptide chain by removing one amino acid at a time. So at a time one amino acid is removed by this enzyme to break down the whatever polypeptide chain, right? Now see, this is the structure of carboxypeptidase A before the substrate binds. So this is the enzyme carboxypeptidase A, right? See what is there. There are many amino acids present, but the important amino acids that we need to look into is that the glutamate amino acid is present at 72 position. The glutamate residue is present at 72 position. Histidine is present at 196 position, and another histidine amino acid residue is present at 69th position. So in this way there are three amino acid residues that are present in carboxypeptidase A. And also take a look at the prostatic group which is present. This is zinc. So this is the structure of the enzyme carboxypeptidase A. Up till now there is no substrate binded to this enzyme, right? Now after substrate binding, after the substrate binds, see what happens. Okay, let me just zoom it. After the substrate binds, what we can see is okay, let us compare both the part. See, this is the structure of carboxypeptidase A, that has glutamate at 72, histidine at 196 and histidine at 69 position. Now this is the structure, the same structure that is here. There is glutamate at 72, histidine at 196 and histidine at 69 with zinc as prosthetic group. Right, now see, after substrate binds, so this red orange structure that you see is the substrate. Right, so the substrate binds to this enzyme. Now as this substrate, this orange part is the substrate, so as this substrate binds to the enzyme carboxypeptidase A, conformational changes take place in the enzyme, which brings glutamate 270, which brings glutamate residue at 270 position, tyrosine residue at 258 position and arginine residue at 145th position. Right, so what is happening? 3 amino acid chains are brought near the active site where the substrate are bound, and this is a conformational change. This is the change in the tertiary structure at the active site, where the enzyme was having only these 3 amino acid residues. Now you can see, after the substrate has bound, other 3 amino acids are brought into picture. These are arginine, tyrosine and glutamate. Glutamate at 270th position, tyrosine at 240th position and arginine at 145th position. So this is called conformational change, brought about only after substrate binding. Right, so we saw that after substrate binding, arginine residue is at 145th position, glutamate residue at 270th position and tyrosine residue at 48th position. Right, so this is the change that comes in the enzyme after the substrate binding, and these have come into picture because these are important in catalytic process. These amino acids play some role in catalytic process by either making the active site more better for the substrate, by making either the active site more fitting better with the substrate. Right, therefore these bringing of the amino acid residues is very important for the catalytic process, and this is what takes place according to the induced fit model. Right, now most of the time induced fit model is the model which is now preferred as for enzyme substrate complex interaction. And also do note that there is a prosthetic group zinc present. Prosthetic group helps enzyme, but these are non-protein. Now what is the catalytic event that is actually taking place? So we already saw that there is initial random collision. This leads to the substrate binding, which induces change in enzyme conformation. So the binding of substrate brings change in the enzyme conformation. This is the process of binding. Right, now this change tightens the fit between enzyme and substrate. So this change is important because this change brings the tightening of the fit between the enzyme and the substrate. So as the substrate gets positioned correctly, bonds are weakened. So the substrate will position correctly, and this will lead to the weakening of the bonds, and this is important for catalytic attack. Right, now this catalytic attack, which means activation, that is the substrate gets activated for the reaction to proceed. Now the catalytic attack leads to formation of temporary covalent bond between substrate and the amino acid that is present in active site. So a temporary covalent bond is formed, and this then leads to breaking down of the substrate. So there is after the completion of binding process, activation process and catalysis process, release of products takes place. Right, so this is the catalytic event which takes place as the enzyme substrate complex is formed. So this model explains somewhat the catalytic event also. So with this we come to the end of the two models. These two models of enzyme substrate interaction are important, and you need to know the difference between both of this. Now we come to the topic of factors affecting enzymes, that is, what are the different factors that affects the enzyme activity. These are temperature, pH, water, concentration of substrate, enzyme concentration, inhibitors, protein poisons, accumulation of end products and radiant energy. So these are the factors, but these are very easy for you to understand. I am sure you are going to understand it very fast, right? So we start with the very first factor, which is temperature. So as we already know now that enzymes are protein in nature, right? So as these are protein in nature, so it is obvious that they would be sensitive to temperature, because proteins are sensitive to temperature. Now the enzyme activity remains optimum at normal body temperature. So when there is normal body temperature, the enzyme activity is optimum. But when there is very low temperature, say for example 0 degrees Celsius, the activity will be minimum. So at very low temperature, the activity would also slow down very much, leading to minimum activity. And when there is increase in temperature, the enzyme activity is going to increase, right? So but this will happen only up to a certain limit, right? So with the increase in temperature, obviously the enzyme activity will increase, but there would be a point when limit will come. So maximum activity is observed at 45 degrees Celsius. So enzymes are maximum active at 45 degrees Celsius, after which the activity gets retarded. So after that the activity will slow down, and at 60 to 70 degrees Celsius the activity is permanently stopped, right? So 45 degree is where maximum activity will take place, and after that, if the temperature increases, the activity retards, and when the temperature reaches 60 to 70, the activity completely stops. So now we come towards the next factor, which is pH. So most enzymes operate effectively at an optimum pH, right? But every enzyme is also specific to a pH requirement. Every enzyme have their own pH requirement, but then enzymes can operate at an optimum pH. This pH is 5 to 9. So at a pH of 5 to 9, most of the enzymes can work good. But there are exceptions where trypsin, an enzyme, this is an enzyme which works in hydrolysis of protein. So trypsin becomes active in alkaline medium. So trypsin requires much alkaline medium to operate properly. Similarly, diastase is an exception which works well in neutral medium, and pepsin is an exception which works well in acidic medium. So there are exceptions, but as I told that every enzyme can operate at an optimum pH of 5 to 9. Now in all these cases, the activity slows down on either side of optimum. So the activity can slow down on either side of optimum. By that I mean, when I say that pepsin is active in acidic pH, so for example pepsin is active at pH 3, right? So if you move more towards lower pH, say for example 1 or 2, it means that the activity will slow down on that side also. And if you move towards basic or neutral pH, say more than 3, that is up to 6 or 7, again the activity can slow down on that side of optimum. So for pepsin, optimum is acidic pH. So on either side of acidic pH, the activity can slow down. Also, extremes of pH value leads to denaturation and inactivation of the protein. So if extreme pH values are there, extreme low or extreme high, then also this can lead to denaturation, that is denaturation of the protein, that is the protein nature will be lost, and also there is inactivation. Now we move towards the third factor, which is water. So in absence of water, always the enzyme activity will get suppressed. So we can take the example of dry seeds. What happens in dry seeds? Dry seeds do not have water. So as they do not have water, the reaction rate is very slow because enzymes are not active, right? So we can take that example. So why this happens? Because hydration is always necessary to carry out the reaction. That is because water provides medium for reaction. So reaction takes place in any medium, and water is the best medium in which reaction takes place. Also, sometimes water molecules themselves are reactants. So therefore water is an important part, and if it is absent, enzyme activity is sure to be suppressed. Now the next factor is concentration of substrate. So concentration of substrate means that how the concentration of substrate can affect the enzyme activity. So with increase in substrate concentration, the enzyme activity will increase, right? So as the substrate concentration will increase, the enzyme activity will increase, but this will happen only up till a point when all the active sites are saturated. So imagine that substrate concentration is increasing, though enzymes are limited. So the substrate, all the substrates will occupy the active sites of the enzyme. So the enzyme can become saturated with the substrates. So after this point, the rate of enzyme reaction becomes steady because there is no active site that can accommodate the substrate, since all the substrate have already occupied the active sites. So as the active sites gets occupied, there is no empty active site, and hence the reaction, the enzyme reaction will become steady, right? So then you cannot see the enzyme activity. So after this point, if you add any substrate, this is not going to have effect on the enzyme reaction. So because there is no active site, and as there is no active site, so no matter what amount of substrate is added, this is not going to increase the activity. So if we want that along with the increase in substrate, enzyme concentration is also to be, if we want, what can be done is that along with the increase in substrate, the enzyme concentration also has to be increased. So as the substrate is increasing, substrate concentration is increasing, enzyme concentration also if increases. So this can give more active sites, and then the reaction can proceed. Now coming to the next part, which is enzyme concentration. So usually what happens is that a very small amount of enzyme is good enough to take large amount of substrates, right? So because enzyme substrate interaction, enzymes can actually carry out processes in very faster rate. So even in seconds, many thousands of reactions are carried out. So this happens because enzymes are efficient enough to take large amount of substrates. right? So a small amount of enzyme is also enough. So increase in enzyme concentration will increase the rate of reaction, because the more you increase the enzyme, the more you are increasing the active site, and this will increase the rate of reaction provided that there is enough substrate. So imagine that enzyme concentration is increasing, so more, it has more number of active site, but if substrate is not there, then what is the point of increasing the rate of reaction, because without the substrate anyhow the reaction is not going to proceed. So it is important that with the increase in enzyme concentration, the substrate must also be present in enough quantity, because two reasons are attributed to it, that higher enzyme concentration will have higher number of active sites, and if there are higher number of active sites and substrates are not enough, then the reaction is not going to take place. And second is that inhibitors may be a problem. So if there are no substrates, then inhibitor will bind to the enzyme active site, and this could be a problem because inhibitors can inhibit the reaction. Then the next is factor is inhibitors. So as the name suggests, inhibitors are those substances that can inhibit the activity of the enzyme, that can stop the activity of the enzyme. Now this can take place partially or completely. So it can either partially inhibit the activity of enzyme, or it can either completely inhibit the activity of enzyme. So inhibitors are mainly classified into two types: competitive inhibitors and non-competitive inhibitors. So as the name suggests, competitive inhibitors are those that would compete with the substrate, right? So what happens, that inhibitors have structural similarity with the substrates, so they are almost similar to the substrate. So as they are similar to the substrate, both of these will compete for the same site, for the same active site. So the substrate as well as inhibitors, since both of them have the same structure, similar structure, they will compete for the same site. And if inhibitors bind first, then this could lead to inhibition of enzyme activity, because enzyme has not received substrate, rather than it has received inhibitor at its active site. But this process is reversible, which means that if we remove the inhibitor, then this could restore the activity. So if inhibitors are removed, the reaction will proceed then normally, as the enzyme will become active by binding of the substrate. So an example that we can take is the conversion of succinic acid, which is the substrate, to product which is fumaric acid. This is done by enzyme succinic dehydrogenase. So this is enzyme, this is substrate, right? So but there is a substance called malonic acid. Malonic acid has similar structure like succinic acid. So sometimes malonic acid can get bind to succinic dehydrogenase, which is the enzyme, and this is an inhibitor. So if malonic acid binds to the enzyme, then the reaction, the product will not be formed, and the enzyme activity is hindered. So this happens because both of these, the malonic and succinic, have almost similar structure. Now non-competitive, as the name suggests, will not compete for the active site, but these are usually poisons. So what these poisons will do, this will destroy the structure of the enzyme. They will destroy the structure of the enzyme in such a way that the change that has happened will become irreversible. So there is permanent irreversible inhibition of the enzyme activity, because the structure of enzyme is itself destroyed. Now coming to the next factor, which is protein poisons. So poisons, as we know, can cause inhibition. So these poisons could be heavy metals like lead, mercury, silver. When these heavy metals are present, this can cause inhibition of the enzyme activity. Then next is accumulation of end products. So we know that at the end of enzyme activity, end products are formed, that is the products are formed, which is the main substance of interest, right? But when there is accumulation of end products, this would lead to inhibition of enzyme activity, because accumulation of end products means that crowding of the product will take place at the active site. Now as the active site will get crowded by product, so this will give less chance to the substrate to bind, and less chance of substrate to bind means that less enzyme activity, right? So when too much of end products are formed, this can get crowded at the active site, leading to the unavailability of the active site for the substrates to bind. The last factor is radiant energy. So it is always harmful because high radiation energy can break bonds in the structure of the enzyme which are not required. So if high radiation energy is given to enzymes, and these can break bonds in the structure of enzymes, and this breaking of bond can lead to structural deformity in the enzyme, and therefore the substrate will not be able to bind, and therefore this will reduce the enzyme activity. Yes, so that is it about factors affecting enzyme. So I hope that it is clear with you, but if you have any doubt, you can always ask me. So with this for today, thank you.